Welcome back to Blood with Miss Hook, Part 3. We are just finishing up talking about white blood cells. So a few white blood cell disorders. Leukopenia, you have not enough white blood cells, a low count. Leukemias, cancers of the white blood cells where they divide abnormally. You can have two types, acute, which involves the early cell divisions, the blast type cells. So you have an early cell division, so everything that gets divided from that early cell is cancerous. So it's really fast and builds up. Whereas chronic is slower, so that would be a lower level cell. And then you have some cancerous cells, but it takes longer for them to show up because there's less coming at one time. And last, platelets. So platelets are these tiny portions of cells. And where they come from are these giant cells called megakaryocytes. And when one megakaryocyte is ready to go, it gets broken into 2,000 to 3,000 pieces. And each of those little pieces is a platelet. The hormone that does this is thrombopoietin. EPO, erythropoietin, was red blood cells. Thrombo, platelets. So they are a lot like white blood cells because megakaryocytes are white blood cells. So they look a lot like little pieces of white blood cells. And they're going to be important for forming what's called the platelet plug when we seal our blood vessels. So the steps to clotting, hemostasis, stopping the bleeding, is first a vascular spasm. So when your blood vessel wall is damaged, the muscle there contracts, bringing the damaged sides closer together so that there's a smaller space for blood to bleed through and a smaller space for us to patch up. Then you have the platelets come to the damaged area and form what's called a platelet plug. And third, coagulation. That's where protein fibers come, trap more platelets and red blood cells, and form the clot. So vascular spasm, vessel wall constricts, brings the edges closer together. Platelet plug, when you open up the connective tissue there, the collagen fibers are exposed, and that's where the platelets stick to. And as they stick, they release these chemical messengers that call in more platelets, which stick, call in more and more. So you have that initial change of calling in platelet cells, and it keeps going in a positive feedback cycle. The signal gets stronger and stronger. And then last, coagulation. So that's where we form the actual. So what happens there is the fibrinogen, that protein that's dissolved in our blood, gets the signal and comes together to form a larger protein that's solid. And that's where we catch the platelets, red blood cells to form the clot. And then, once the clot forms, it tightens, pulls the vessel walls together, and you're done. Vitamin K is needed. It helps with clotting factors. And then if you do have any unwanted clots, they're usually dissolved by plasmin. So this is a nice picture showing how the fibrinogen is solidifying into fibrin, catching the red blood cells, and forming the clot. So I would pause the video, review the steps of forming a clot. So let's look a little bit more at how we turn fibrinogen into fibrin. The most important thing here is thrombin. So thrombin is released, and that's what causes these little Q-tip-like proteins to form together into a larger molecule that solidifies and forms the clot. Anytime you have a clot form where it shouldn't be, where the vessel isn't broken, it's called a thrombus. And if that thrombus breaks free, it's called an embolus. So there's an embolus flowing through the blood. And this can be dangerous. If you have a narrow pathway there, perhaps from the buildup of plaque, you can have blood flow restricted. That's called an embolism. So a pulmonary embolism would happen in the lungs. It would be a heart attack in your heart, a stroke in your brain. So people at risk will take different medications. So like warfarin, That example is shown here, where it doesn't allow you to make vitamin K, so you don't make as many clotting factors. Aspirin reduces the ability to spasm your blood vessel, as well as decreasing platelet plug formation. And heparin stops thrombin, so it stops the fibrinogen converting to the fibrin. Bleeding disorders. If you don't have enough platelets, that's thrombocytopenia. So there your ability to clot is reduced. Same idea with hemophilias. So these are genetic, hereditary, so you can inherit them. And it's inherited on the X chromosome. And if you have a mutation, you can't properly clot. It can be deadly. You'll see this especially in males because they only get one copy of the X gene. So if they get a bad copy, they're out of luck, whereas females have 2 chances. Royalty in Europe inbreeds a lot, especially a few hundred years ago, and they had hemophilia. And they kept producing princes with hemophilia who would die young because they couldn't properly clot and they weren't bringing in healthy genes. You can give someone a transfusion of blood if they have anemia or other issues with their blood. But you have to make sure you have the right blood types. Otherwise, they can be fatal. So we're going to look at what that means. So human blood groups are proteins. They're on the outside of your red blood cells. And basically, you have to get blood from people who have proteins that your body doesn't interact with, doesn't respond to. The ones that are in the common population and not just in like a single family are the ABO blood type group and the RH. So first let's talk about the ABO. In ABO, you have a gene, one copy from your mom and one from your dad. And those genes can either code for an A protein, a B protein, or they can be mutated and not code for any protein. In that way, you're O. So if you get an A copy and a B, one from your mom, one from your dad, you're AB. If you get B from one, you could get B from both your type B. You could get a mutated one that doesn't do anything, and then a copy of B, you would be B. What's really neat is based on what proteins you have on your red blood cells, your body makes antibodies against any of the proteins you don't have. So if you have type blood O, you don't have any proteins. So your body makes antibodies against A and B. Whereas if you're type AB, you have both proteins, so your body doesn't make any antibodies at all. So if type O blood with these antibodies saw this red blood cell, these would attach, like this could bind to the A antigen of one red blood cell and another, and that could attach to another, and you could have this clotting occurring. Type B, they make antibodies against A, and A makes antibodies against B. So whatever you don't have, you make antibodies against. So you have to make sure if you get blood, you don't get blood with the proteins that you're making antibodies against. So that's why type O is the universal donor. It doesn't have any proteins on the outside. It won't react in anyone's blood. Whereas type AB is the universal recipient because it doesn't have antibodies. It won't attach to any of these red blood cells. So there's a table showing how likely different blood types are based on descent. Here's a nice picture showing what happens if you have a protein on the outside and are given that antibody. So let's say this is protein A, and this is anti-A antibody. If they come in contact, the antibodies stick to red blood cells and form a clumped population. So here, this would be like type A blood given anti-A antibodies. This blood, if O, for example, is given anti-A antibodies, nothing's going to happen because it doesn't have the A protein on the outside, so it's not going to react. The other type of blood group that we look at is Rh. It's named for rhesus monkeys because that's where they found it first. So if you're Rh positive, you have that protein on the outside. And if you're Rh negative, you don't have it on the outside. So O negative doesn't have any proteins, whereas AB positive has all three. The neat thing about Rh is that we don't naturally make antibodies against it. So if you came in contact with Rh positive and you were Rh negative, the first time, nothing would happen. But the second time, your body has learned about this. It's now making antibodies against it and would react. A time where that's really important is during childbirth. So pregnant women who are Rh negative, they don't have the proteins. If they have a baby that's Rh positive and they're exposed to that blood, their body's going to start making antibodies against it. So if they have another baby, those antibodies attack it. So what we do instead is we give them what's called RhoGAM serum. And basically, it tells the body that they're making the antibodies and that it doesn't need to learn and do it itself. And lastly, let's look at some diagnostic blood tests. So our blood can tell us a lot about our body's function, especially if we can compare it to urine. But blood tests, hematocrit, percent that's red blood cells. You can look at glucose levels. You can look at the blood cells under a microscope. Look at how big the red blood cells are, if they're mal-shaped, how many there are. You can also look at the different types of white blood cells. See which kinds are elevated to determine what's going on in your body. You can look at prothrombin and platelet counts to look at clotting ability. You can do a whole blood chemistry profile or a complete blood count, where they look at everything we looked at in the case study on anemia. Red blood cells, white blood cells, and platelets, so you know a bit about everything. Okay, that's it. Have a great day.